everyone. My name is Amy Smith, and I'm from Woods Hole Oceanographic Institution. And today I'm going to talk to you about Lohi Seamount and its potential for an scientific analog to Enceladus or other ocean worlds that might have, hi Vincent, that might have some applications to hydrothermal vents on other ocean worlds. So I have a lot of co-authors here because this is a big collaboration between NASA, NOAA, OET, Woods Hole, and some other universities. And then there were some other older samples that I used that I'm going to talk about today as well from Lohi Seamount that were part of the FEMO project. So let me get started. So what you're looking at right now is actually a hydrothermal vent. I know it might not look to you like what you've seen or imagine a hydrothermal vent to be. It's rather small, but the temperatures of the fluids coming out of this vent are about 30 to 40 degrees Celsius. And you can see that there are a lot of orangey yellow fluff around. That's actually a bacterial mat. Mostly it's made up of this organism called Microbes feroxidans, which is a deep subsurface organism that oxidizes iron and fixes carbon. And so these organisms characterize this environment. You can see them all over the rocks at the top of this volcano where you have these hydrothermal vents. And this actually, we named Dragon's Cave because it kind of does look like it has dragon's eggs inside the cave. So that's one of the places that I'll talk to you about today. So there are lots of ocean worlds in our solar system, and besides Earth, my favorite one is Enceladus because we have evidence of hydrogen production and there might be hydrothermal vents on Enceladus. So the reason why we went to Lohi Seamount is because we think it might be a good analog for this type of environment, mainly because this hydrothermal vent system is not on a spreading ridge, which is where you find most of them. It's actually an intraplate hotspot volcano, and because we don't expect for plate tectonics to exist on these other worlds, this is probably more likely like what we would find. So at Lohi Seamount, we have hydrothermal venting. It's a hotspot volcano. There's no sunlight because it's about 1,000 meters below the surface. And there are water rock reactions to drive chemosynthesis. And these organisms at the surface that you see in the mat here, this is one of those mats of Microbes feroxidans. Those are actually chemosynthetic organisms as well. And then this, this blue dots here, those are actually microbes in fluids from Lohi Seamount. If you can stain the DNA of the microbes and look at it under the microscope, and that's what they look like. So this is a map of Hawaii here, and then you can see this is the hotspot that created all the Hawaiian islands, and Lohi is the next island that's being formed underneath the ocean right next to the big island. So what makes Lohi Seamount interesting is that it's very different from on-axis vent sites, as I mentioned. It's also a low temperature site, so the temperatures of the fluids are around 30 to 40 degrees Celsius. At some point they were a little bit higher, sometimes they're lower. But what's interesting also is that there's no sulfide in the fluids. There's a lot of iron and CO2, and there's a little bit of hydrogen. It's below the detection limit, which is about 3 micromolar or less. But still, it's possible that microbes could use it for energy. We don't see very many like typical vent organisms at this site. We only really see those mats, a couple of shrimp, and a few fish. A previous paper by Jason Sylvan et al. In 2017 said that nitrogen, sulfur, and iron metabolisms are probably likely in the fluids that are coming out of the vents. But at that time, really, it's just energy modeling, and he had some taxonomy that he reported. And then I have taken those fluids and looked more deeply at the metagenomes, and I'll present that as well today. So as I mentioned before, this location is dominated by these iron mats or Microbes. But one interesting thing I want to point out is that they have to have oxygen. And whether or not there's oxygen on some of these other ocean worlds is up for debate. So we're not really sure if this is a good location or not based on that. But there are other ways to oxidize iron, so it's possible that this type of organism could exist. So our main question with this study was to look at what life looks like in the subsurface, not necessarily focusing on those iron mats that have been studied for years and years. So what I did on a recent cruise in August and September of last year was to take some fluid samples from several different sites on the Lohi Seamount and look at cell densities to get an idea of how much energy might be in the system and how much biomass can be supported by that. Look at the taxonomy again, do metagenome studies, and then to culture some organisms. And you can notice here, this is also another vent site that we were sampling. This is called a super sampler that we use to sample the fluids that are coming out of the rock. Also, I'll be talking briefly about the last bit of our cruise. We were able to go and sample some not vent fluids necessarily, but some fluids from the Kilauea ocean entry site. If you remember, in August of last year, Kilauea had just been erupting for a period of time for several weeks, a couple months. So we went to that site at the very end of our cruise to look at the ocean entry to see if we could see any life there that had colonized the basalt that had gotten into the ocean. I would also like to point out that during our cruise that was 3 weeks, there were 4 hurricanes. So the first hurricane was Hurricane Lane, and it kind of hit that big island pretty hard. So I'm going to show you a video of what we encountered on the ocean entry site, and it doesn't look like fresh basalt because it's covered in sediment and there are like palm trees and stuff everywhere. But that's what it is, trust me. Okay, so this is 635 meters down at the Kilauea ocean entry site, and you might recognize something kind of blowing in the wind, that orangey yellow looking microbial mat there. So we got a sample of the mat, and then we're sticking our super sampler in the different holes to see if we could find any fluid flow. And we actually measured a elevation of temperature of 0.8 degrees above ambient. So just a little bit warmer, but it was still warm after 3 weeks of inactivity from the volcano. So that was pretty exciting to us. And it was also really exciting that we saw life had already colonized there, and it looks a lot like what we saw at Lohi Seamount, which is not far away. So I also have a couple of videos of some really cool microbes that I was able to produce in culture on the ship while we were there. The first one is an iron reducer enrichment, which grew pretty well. And then the other is a rock that we took from an old vent that was at the top of Lohi Seamount. But you can see there are a lot of sulfides, and it's quite beautiful, and I just couldn't stop staring at it, and I didn't want to give that rock away, but I had to. And then these are some of the other beautiful rocks that we were able to collect from the summit. Hopefully it will advance. Okay, so how am I on time? Okay. So now I want to talk to you about the cell density in these fluids and how it compares to some other sites as far as whether or not they're hydrothermal vents or bottom seawater. So if you can see, all of the orange bars here are cell counts from Lohi Seamount. The first set is from the 1980s that were reported right after an eruption. And then these here are the ones from the 2018 sampling site that we took. And then this is that Mustard Hollow, is what we're calling it, the Kilauea ocean entry site. So those fluids that were 0.8 degrees above ambient. So those are all the cell densities here. And then if you follow this line, that will show you where Lohi comes out on this graph. All of the light blue are bottom seawater samples. Whether or not they're close to Lohi or in another location, they all kind of are a little bit lower than Lohi, but Lohi in general is kind of in between what we might find in bottom seawater and a sulfide-rich hydrothermal vent such as this one, which is Mariana Back Arc here. So you can see those are much higher cell densities And those also support a large amount of macrofauna and lots of other life forms as well. So Lohi looks like it's kind of a lower energy hydrothermal vent system. So this is the 2018 data that we got. The first top bar is the bottom seawater. Then we have that Mustard Hollow, which is the ocean entry site. And then these are some of the different vents that we sampled at Lohi. And this is just looking at the taxonomy from a metagenome or metagenomes that I have been producing in the lab that are now ready to be sequenced, but we wanted to check the taxonomy first. And you can see actually anything that's yellow or orange, I've tried to color code with, that's a Mare profundus, so that same organism that you find in the mat at the surface, okay? So we've got 3 different types of Mare profundus, which are the most abundant organisms here. And then even in that Mustard Hollow site. So that mat that looked like it might have been one of those types of organisms actually was. So the same organisms that colonize the vent sites at Lohi are actually colonizing new lava flows that are coming into the ocean at the Kilauea volcano site as well. And then we have some other organisms that are unknown or grossly unknown, I guess. So hopefully the metagenomes that I produce later will give us more information about what these organisms are doing there and whether or not they're actually doing what we think they are. And so now I'm looking at 2006 data. So this is these are metagenomes from some samples that were taken in 2006 at the same locations. So we have, again, bottom seawater, and then these are 3 of the different vent sites. And you can see based on, this is just carbon fixation and the amount of genes that were present in each of the metagenomes, and then which pathways are that these are present in is color-coded. And you can note for sure right away that you have some yellow and pinks here that are present in the Lohi Seamount samples that are not present in the bottom seawater, and those are anaerobic carbon fixation pathways. So the ones in the bottom seawater are mostly Calvin cycle and some reductive TCA cycle as well. But once you get to the Lohi fluids a little bit deeper and there's more anaerobic fluid coming out, then you start seeing anaerobic carbon fixation as well. So we're kind of sampling that deeper subsurface organisms. And then this chart is just a summary of those carbon fixation pathways, the nitrogen metabolisms and sulfur metabolisms, and then comparing those to the bottom seawater sample. So this is the same that I just showed you just a second ago, but I do want to point out that there's no methanogenesis potentially occurring in these sites. We do have some genes in methanogenesis, but there were no complete pathways out of a whole metagenome. We don't have all of the genes. So that's something to keep in mind. And then you can see here there's a shift from the Calvin cycle and bottom seawater to these more anaerobic carbon fixation pathways in the Lohi vent fluids. Nitrogen metabolism is really, there's a higher potential for that as well in these Lohi fluids. And then the sulfur metabolism, both bottom seawater and Lohi fluids have a high potential for that as well. Okay. I just want to point out, too, that there are a lot of hydrogenases present in these fluids. Even though there is not a lot of hydrogen, there's a high potential for hydrogen utilization. It has been pointed out to me that Mare profundus has a lot of these hydrogenases, so I think it would be interesting to look and see if those are the organisms that are using these as well. So our preliminary results are that I have metagenomes that show potential for anaerobic carbon fixation and hydrogenotrophy in this Lohi vent fluid community, including the reductive TCA and Wood-Longdahl pathway for acetogenesis, not methanogenesis. That we have nitrogen and sulfur cycling that's potentially important in this community. The cell density is really low. We do have iron oxidizing bacteria present, but it's not the majority of the community like we have in the mats. And then I'm culturing organisms in the lab based on organisms that grow on minerals like olivine, iron, and hydrogen, and trying to determine who the autotrophs of the community are, who's fixing carbon. And then I have some more experiments coming as well. So for ocean worlds, This Lohi site, it is a low energy system, and there is evidence that there's oxygenated seawater circulating within the crust, and that's being captured in that community. So perhaps going deeper, we might be able to get a more pure sample for ocean worlds. And we already see evidence of anaerobic carbon fixation pathways, and then we have some unusual organisms potentially using them. So with that, I would say thank you.